Good evening to your students. So, we have completed amine chapter in the last class. Okay. So today we are going to start with the next chapter, that is biomolecules. Okay, we are going to discuss about biomolecules. Can anybody tell me what do you mean by biomolecules? Yes. Can anybody tell me what do you mean by biomolecules, students? Yes. So biomolecules, can you give any examples of biomolecules? Is it necessary for the living organism? Yes. Is it necessary for the living organism? Are these biomolecules necessary? Yes. So what are these biomolecules? Okay. So biomolecules, they are the organic substance. Okay. They are the organic substance which are used to build up our living organism. Okay. And why they are required? They are required for the growth. Okay. For the growth and for the maintenance. Okay. So the biomolecules are required for the growth and maintenance of this living organism. Okay. So which are the different examples? So which are the different examples like carbohydrates, protein, nucleic acid, right? Your lipids, right? So these are all the examples of biomolecules. So what do you mean by biomolecules? So these are the organic substance. Okay, these are the organic substance which are used to build up the living organisms and they are required for their growth and maintenance. Okay, so examples: carbohydrate, proteins, nucleic acid, lipids, etc. Okay, so there is something called as biochemistry. Okay, so the branch, okay, that is a branch of chemistry, okay, which is dealing with the structure, functions, and the interaction of biomolecules. Okay, then this it is called as biochemistry. Understood? So the branch of chemistry which deals with the structures, functions, and the interactions, okay, of the biomolecules is called as what? Biochemistry. Understood? Okay. Okay. So let us start. So the first biomolecules. So yes. Okay. So what are the biomolecules? So just now I've told you, which is present in the living organism. Okay. Yes. So these biomolecules, they are nothing but they are predominantly they are made up of carbon. Okay. So carbon is the main element which is present in the all these biomolecules. So generally these biomolecules are inorganic and organic. So organic means what? Which is made up of carbon compound. That is, they are carbohydrate, lipids, amino acid, proteins, enzyme, nucleotides, nucleic acid, and vitamins. Okay. So we are going to discuss about the what we are going to discuss about the carbohydrates. So let us discuss about the carbohydrates. So what do you mean by carbohydrates? Yes, anybody? What do you mean by carbohydrate? Yes. So these are the hydrates of carbon. Okay, what are carbohydrates? These are the hydrates of carbon. Okay, so you write the general formula as CN. Okay, you write the general formula as CnH2ON. Okay, what is the general formula? CnH2ON. So N can be your any number. Okay, any number of molecules is which are associated. So carbohydrates. Okay, these are the hydrates of carbon and they have the general formula of CnH2N. So what can be the examples of carbohydrate, students? So I can give the examples of carbohydrates as glucose. Okay, the formula is C6H12O6. Glucose is what? C6H12O6. Fructose, that is C6H12O6. Again, I can give you sucrose. Okay, that is C12H22O11. Okay, so these are some of the sugars. Okay, that is, these are some of the carbohydrates. Okay, so there are There are certain carbohydrates, okay, which will not fit in the formula, okay, like your ramnose and your this what ramnose, okay, so that is a that is C6H12 O5. So this is a carbohydrate which will not fit in the molecular formula of CnH2ON. Okay, so the what is the definition of carbohydrates? So basically, the chemically, when you give the definition of carbohydrates, so they are the optically active polyhydroxy aldehyde or ketone. Okay, what are these? They are the optically active polyhydroxy aldehydes or ketones. Okay, so which produces, okay, the compounds which produces such units upon hydrolysis. Okay, so these are the, what is the definition? Please remember. So carbohydrates are the optically active polyhydroxy aldehyde or ketones. Okay. Yes. So next is classification of carbohydrates. Okay. Next is what? Classification of carbohydrates. Okay. So based on the number, okay, how much simpler units it is giving. Okay. After hydrolysis, the carbohydrates, how much simple units it is giving. Okay. So that is called as what? That is called as, we are classified based on that. Okay, so they are basically categorized into three. Okay, that is, one is the monosaccharide, then oligosaccharides and polysaccharide. So let us see what do you mean by monosaccharide, oligosaccharide, and polysaccharide. Okay, so what do you mean by monosaccharide? So this is a first classification. Okay, so this is a simplest hydrocarbon. Okay, it is a simplest hydrocarbon, okay, which cannot be further hydrolysis. It cannot undergo further hydrolysis. Okay, so okay, so what can be the example? So I'll give the example like your glucose, okay, then the fructose, okay, then you have ribose, okay, and galactose, etc. So these are some of the examples of the monosaccharides. So monosaccharides, so these are the simplest carbohydrates, okay, which cannot be further hydrolyzed. Okay, so the formula is CnH2n ON. Okay, CnH2nON. So this is the general formula. Okay. Then next is oligosaccharide. Okay, what do you mean by oligosaccharide? So oligo means few. Okay, oligo means oligo. Oligo word oligo means few. So these are the carbohydrates which on hydrolysis will give you 2 to 10 monosaccharides. Okay, they will give you 2 to 10 monosaccharide units. Then they are called as a oligosaccharide. Okay, so carbohydrates which will give you 2 to 10 monosaccharide units, they are called as what? They are called as the oligosaccharides. So example can be sucrose, then maltose, okay, then lactose, okay, then I'll give you raffinose. So these are some of the example of oligosaccharide. So these oligosaccharides, so they are further classified as disaccharide, trisaccharides, and tetrasaccharide. Di means 2, tri means 3, and tetra means 4. Okay, depending on how many monosaccharide units they have given. So they have as di, tri, and tetra. Okay, next is polysaccharide. So by the word poly itself, you will come to know that on when the carbohydrates undergo hydrolysis, they will give you many or large number of monosaccharide unit. Okay, they will give you large number of monosaccharide unit. So what are the example? Starch, cellulose. Okay, then you can give glycogen. Okay, etc. So these are some of the examples of your polysaccharide. Is it clear about carbohydrates? So what you need to know about carbohydrate? They are the optically active polyhydroxy aldehydes or ketone. Okay, carbohydrates on hydrolysis will give you monosaccharide units. Okay, they'll give you units. So these carbohydrate they are classified as monosaccharide or oligosaccharide and poly. Mono means these they are the simplest carbohydrates which cannot undergo hydrolysis. Okay, further we are given the examples. Okay, then oligo means few. So these are the carbohydrates which on hydrolysis will give you 2 to 10 monosaccharide units. Okay, depending on how many monosaccharide units are there. Okay, so it is given. Then poly means more. So large number of monosaccharide units are given. Okay. Yes. So now this carbohydrates, they are further classified as reducing and non-reducing sugars. Okay. They are what? They are classified as reducing and non-reducing sugars. Now, what do you mean by reducing and non-reducing sugars? Okay, reducing means what? These carbohydrates can. Okay, so I'll take a just a second. Okay, so what do you mean by reducing and non-reducing? So further, the carbohydrates are classified as reducing sugars. They're classified as reducing sugars and non-reducing sugars. Okay. Now, what do you mean by the word reducing and non-reducing sugars? So, reducing means, okay, reducing means these carbohydrates, they reduce Fehling solution and tolerance reagent. Okay. So, these carbohydrates, so here these carbohydrates, what they do? They they reduce the Fehling solution. Okay, the reduced Fehling solutions and tolerance reagent. Okay, and tolerance reagent. Understood? And non-reducing means what? Okay, these carbohydrates. Okay, these carbohydrates. What they do? They do not reduce. They do not reduce Fehling solution. Okay, Fehling solution and tolerance reagent. So this definition you need to remember. Okay, they do not reduce and they reduces the Fehling solution and tolerance reagent. Now, what are the examples? Okay, so here for reducing agent, all monosaccharide units. Okay, all monosaccharides and few disaccharides. Okay, few disaccharides. So which are the disaccharide units? Like your maltose and lactose. So they will give you the test for reducing. Non-reducing, which will be the example? It is sucrose. Okay, sucrose is the example of a non-reducing sugar. Is it clear? Yes. Is it clear? Okay. So what you have to remember? So what are reducing? What are non-reducing? Okay. Next, when we are discussing about the what is that? What do we call? About the monosaccharide unit. Okay. When we are discussing about the monosaccharide unit. Okay. When we are discussing about the monosaccharide unit. So just see here. Okay, so this monosaccharide unit, okay, what we are talking about, when they are with aldehyde group, okay, they are called as aldoses. What they are called as? They are called as aldoses. So in monosaccharide unit, if you have the functional group aldehyde, then we call it as aldoses. And if we have ketone, okay, if we have ketone present, then we call it as ketosis. What we call? We called it as ketosis. So monosaccharide unit with an aldehyde group are called as aldoses, while the ketone while the monosaccharide unit with ketone group, that is ketonic group, it is called as ketosis. Okay, now based on the number of carbon atom, this monosaccharide units they are called as. So if you have 3, then they are called as trioses. Okay, 4 tetrose, 5 pentose, 6 hexose, 7 heptose. Okay, so based on which functional group it is having, if it has a aldehyde, so it is aldotriose. Okay, if it has a ketone, so ketotriose. So like that. So aldotetrose, ketotetrose, aldopentose, ketopentose, aldohexose, ketohexose, aldoheptose, ketoheptose. Okay, like that. So let us take an example. So first example of a this is glucose. Okay, we are going to see about glucose. So let us see about glucose now. Okay, so glucose, it is where it is. It is called as a dextrose sugar. Okay, glucose. The name is grape sugar. It is called as a grape sugar. Okay, and it is dextrose present in. So it is a dextrose which is present in sweet fruits. Okay, it is present in sweet fruits and honey. Okay, it is present in sweet fruits and honey. Okay, so what is the preparation of glucose? So how do we prepare? Okay, preparation of glucose. That is, first is from cane sugar. Cane sugar is nothing but your sucrose. Okay, from sucrose. So how you can do? So this sucrose, that is your C12, H22O11, it undergoes hydrolysis. Okay, acid hydrolysis in the presence of acid. Okay, sucrose on acid hydrolysis in the presence of dilute HCl. Okay, or H2SO4 in alcoholic solution will give you glucose and fructose. Okay, it will give you what? Glucose and fructose. So this is glucose and this is fructose. So like this, you can prepare your glucose. So this is sucrose. So from sucrose in acidic condition, that is on acid hydrolysis, will give you what? Will give you glucose and fructose. Okay. So from this, we can prepare our glucose. So another reaction from starch. Okay, from starch also we can prepare, that is take glucose. Okay, how we can do? By starch. So starch is C6H10O5. Okay, so when starch, we do hydrolysis again. So we are doing a hydrolysis, acid hydrolysis at 393 Kelvin. Okay, and 2 to 3 atmospheric pressure. I am going to get what I am going to get. I'm going to get glucose. Okay, I'm going to get glucose. So another what method of preparation? Is it clear? Okay. So now we are going to see the structure elucidation of glucose. Okay, how we are going to see the structure of glucose. So experimentally, okay, the glucose has an open structure. Okay, it has an open chain structure. So according to the mass determination, the molecular formula is C6H12O6. Okay, glucose has the molecular formula of C6H12O6. Okay, so there are 6 carbon atoms. Okay, so these 6 carbon atoms, they are in straight chain. So how you are going to give the reaction? So glucose, okay, you have to heat it with Hi, that is hydrogen iodide. So what I am going to get? CH3, CH2, 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 CH2 and CH3. Basically, it is a N-hexane. So N-hexane, what it tells you, that the number of carbon atoms are for 6. So 6 means in the glucose structure also there are 6 what? There are 6 uh, carbon atoms. Okay, there are 6 carbon atoms. So this is told here. Okay, so just a second. Yes. So see what is there? So we have glucose. So glucose on heating with Hi. Okay, what I'm going to get? N-hexane. Okay, I'm going to get N-hexane. So this is the first structure which tell you that there are 6 carbon atoms in the glucose structure. Now next is we have to see for carbonyl group. Next is we have to see for carbonyl group. So what we have? We have a structure of glucose. Okay, so glucose when you react, okay, when you react it with uh, so CHOH 4 times. Okay, and CH2OH. So this glucose will react with hydroxylamine. What is hydroxylamine? NH4 hydroxylamine. Okay, so how you are going to NH2OH. Okay, so what we are going to do here? So we are going to react it with hydroxylamine. Okay, and what I am going to get? What I am going to get? So I am going to get a oxime. I am going to get what? Oxime formation. Okay, I am going to get a oxime formation. So only at the carbonyl carbon, the reaction happens and rest all things remain the same. Okay, and also we can add a molecule of hydrogen cyanide. Okay, when you add molecule of hydrogen cyanide, it forms cyanohydrine. What it forms? It forms cyanohydrine. So there will be addition of CN and OH. So rest all things will remain the same. Okay, rest all things will remain the same. So like this. Okay, so this will give you glucose cyanohydrine. This will give you glucose cyanohydrine, and this is glyoxime. This is glyoxime. So what this reaction tells you? This indicates that there is a presence of carbonyl group. This indicates that there is a presence of carbonyl group. Is it clear? Is it clear? Okay. Fine. So second reactions, what it tells you when you react it with hydroxylamine, so there will be formation of this oxime. Okay. And here when it reacts with hydrogen cyanide, I'm going to react, we are going to get a cyanohydrine. Okay. So glucose cyanohydrine. So both tells you what? There is a presence of carbonyl group. Okay, carbonyl group. Then third reaction, we have to see whether the carbonyl group is aldehyde or ketone. So what we are doing? So again, we are going to take the glucose. Okay, I'm going to take the glucose, and what I'm going to do? I'm going to do oxidation. How we are going to do oxidation? With bromine water. Okay, with bromine water. So when you do bromine water, only the carbon is going to get oxidized to acid part, and rest all things remain the same. So what it shows that the glucose on oxidation with bromine water, okay, is giving you glyconic acid. The name is glyconic acid. Okay, the name is glyconic acid. Is it clear? So it gives you glyconic acid. So this indicates that the carbonyl group is a aldehydic group. Okay. Next is we have to see for the presence of 5 OH group. Okay. There are 5 OH groups in this. So how we are going to determine that? So again, take the glucose. Okay. Take the glucose CH2OH. Then what I am going to do? I am going to react it with acetic anhydride. Okay. I am going to react it with acetic anhydride. So what I am going to get? So all the OH. So wherever OH are there, so there your acetyl group will added. Okay, it will be added, and what I am going to get? I am going to get the, what is the compound? It is glucose pentaacetate. Okay, glucose pentaacetate. So this indicates that there are 5 stable OH group. So there are 5 stable OH group. Okay, so your 4 and your 5. Okay, so 4 and your 1, 5. So there are presence of 5 OH groups. So this is a structure elucidation. We are doing the structure elucidation. Is it clear up to here? Okay. Let us go for the next one. So next is the presence of primary alcoholic group. So what I am going to do? So again, so I am going to take the, I am going to get the glucose. Okay, I'm going to take the glucose, do oxidation. Now here, this time I'm going to take what? I'm going to take HNO3. Okay, HNO3. So what will happen? Both will turn COH. Okay, both is going to convert into acid. So that is a saccaric acid. Okay, this is a saccaric acid. This saccaric acid, okay, so from gluconic acid also you can get saccaric acid. So this indicate that what? There is a presence of one primary alcohol. Okay, so based on all this, what you will come to know, that there is a open structure of the glucose, and the structure of glucose is like this. How it is? CH double bond O. From all this, okay, we are going to get the structure of glucose, and the structure of glucose is like this. Okay, so this is a glucose structure. So 1, 2, 3, 4, 5, and 6. Understood? So this is the open chain structure of glucose. Okay. So next, there is something called as D and L form. Okay, D, when we use D, capital D and L configuration, okay, there is something called as D and L configuration. So when we use it, so to assign the relative configuration. Okay, now suppose I have a compound like this. Okay, HOH, CH2OH, then CHO, HO, H, CH2OH. So you can see here it is at right hand side OH and here it is at LHS. Okay, so these are the standard reference. This is a compound called glyceraldehyde which is taken as standard reference. So this is called as a D form when the OH is on the right hand side, and when the OH is on the left hand side, then it is called as L form. Okay, yes. Then what is the cyclic structure of glucose? Yes, how do you give the cyclic structure of glucose? So this is so you have the alpha and beta in the form of hemiacetals, right? So we have the hemiacetal formation. So let me write the hemiacetal formation of the glucose. Okay. Okay. So now here it is. Okay. So this is the alpha D glucose structure and this is the beta D glucose structure. Okay. So this is the alpha D and beta D structures. So what you can see, so this is a open, so here this is called as a hemiacetal formation. Okay, in the hemiacetal formation, the structure is proposed, okay, of the glucose. Okay, yes. So based on this, we can write the Hayward structures also. So let me write the Hayward structure. So it is same. Okay, so with this, with respect to the hemiacetals only, we are writing the Hayward structure. So Hayward structure, so it will be in the ring form. Okay, it is in the cyclic structure form. So this is HOH, HOH, then OH, H, HOH, CH2OH. So I'll show you this is for alpha D glucopyranose. Okay, because it is a pyranose structure. So it is called as the alpha D glucopyranose. So when I see the numbering, here where you can see the numbering alpha D glucopyranose. So 1, 2, 3, 4, 5, and 6. So similarly, this also is 1, okay, 1 carbon, second, third, fourth, fifth, okay, and sixth, like that. Understood? Okay, so like this, you're going to number. So similarly, with respect to that, we are going to write the beta also. What is the change in alpha and beta? Here, this will be OH will be up, okay, and rest all things will be same. Is it clear? Okay. So like this. So rest all things will be the same. Only the change in the OH at first position. Okay. So when I number this, so I'll number it from 1, 2, 3, 4, 5, and 6. So this is what? This is beta D pyranose, glucopyranose structure. Okay. Beta D glucopyranose structure. Is it clear? Okay. So next we are going to discuss about fructose. Okay, so fructose, how we are obtaining fructose? So fructose also we are obtaining it from hydrolysis of sucrose. Okay, so it is a it is what? It is a ketohexose. What it is? It is called as a ketohexose. So fructose is called as ketohexose. Okay, yes. So, so ketohexose, it is called as a ketohexose. So when you see the structure, so here is the structure of fructose. Here you can see the structure. Yes, so this is a, here in the ketohexose you can see this. So this is the structure of the fructose. Okay. So fructose, so it has both levorotatory and levorotatory. Okay. And so this is also will give you a hemiacetal form. Okay. And when you see the Hayward structure, so this is the Hayward structure. So based on this, we have to write first the hemiacetal formation. Okay, we are going to write the first the hemiacetal formation. So let me take a new slide. Okay, so here you can see. So how we are supposed to write the structure. Okay, please understand this. So let me write it here. So this is CH2OH. Okay, so you have a keto group. Okay, then you have OH. H, okay, then H, OH, then H, OH, and CH2OH. So this is 1, 2, 3, 4, 5, and 6 carbon. So if I write the hemiacetal formation, okay, if I write the hemiacetal formation, so how it will be, so CH2OH, OH, then COH. So this is your alpha D fructose. Okay, this is your alpha D fructose. Let us write the beta D fructose. Okay, let us write the beta D fructose. So this is going to, so like this. Okay, so now here also how it will be. So HO. So only the position will be changed at the first carbon. Okay, first and second carbon here. First, second, third, fourth, fifth, and sixth. So here also you can see. So this is CHOH, CHOH, CH, CH2OH. Okay, so like this. So this is beta D fructose. Okay, it is beta D fructose. Fine. Okay. So these are the Hayward structure, hemiacetal formation structures. Okay. Now we'll write the Hayward structure. So based on this only, we'll write the Hayward structure. So this is a Hayward structure for your alpha D fructose, fructofuranose. So this is called as fructofuranose. Okay. So here you can see, so A OH, H, HOH, then CH2OH and H. So first, second, third, fourth, fifth, and sixth. So this is what? This is alpha D fructofuranose. Furanose, okay, it is a furanose structure. Then we will see here beta D. Okay, let us see beta D furanose. So here I'll write, okay. Okay, so let us write, okay, so here it is OH, CH2OH. So rest all things will remain same. Okay, so H, this is HOH and CH2OH and H. So when I put the numbering, so it will be 1, 2, 3, 4, 5, and 6. So again, this is also beta D fructofuranose. Okay. Yes. So next we are going to, so these were the examples of the monosaccharides. Okay, glucose and fructose. So we are seeing the examples of monosaccharide. Okay. So glucose and fructose we have seen. Now next is disaccharides. So disaccharide is what? How we get disaccharide? When two monosaccharide unit they get condensed. Okay. Or you can say when two monosaccharide unit they are joined together. So there should be one linkage. So the linkage which is present in a disaccharide is glycosidic linkage. Okay. It is a glycosidic linkage which is present in a disaccharide. So the example is maltose. So we'll take a maltose, which is also called as a malt sugar. It is called as the malt sugar, that is C12H22O11. Okay. So it is composed of alpha D glucose unit. It consists of two alpha D glucose units. So how is the structure? So this is a structure. Okay, here you can see this is the structure. Okay, this is a structure. And where is the alpha linkage present? So if I number it, so this is 1, 2, 3, 4 carbon, 5 and 6 carbon. Okay. Similarly 1, 3, 4, 5 and 6 carbon. So the linkage is present in C1 and 4. So C1 of one glucose and C4 of another glucose, the glycosidic linkage is present. Okay. Yes. So what is maltose? Maltose is made up of two glucose units. Okay. Next is sucrose. Okay. Next is sucrose. So when we see sucrose structure, what is sucrose called as? Sucrose is called as the cane sugar. Okay. It is also called as cane sugar. C12H22O11. Okay. So it is made up of what? It is made up of alpha D glucose and beta D fructose. Okay, so the structure is like this. So you have to practice this all Hayward structure. Okay, this Hayward structures are very, very important. Okay, the Hayward structure is very, very, very important. So this can be asked for 2 marks. Any they'll ask. Okay, any one of this. So CH2OH, OH, H, H, OH, CH2OH, H. So if I number, okay, so it is present. So 1, 2, 3, 4, 5, and 6. And this is what? 1 and if I give to number, okay. So C1, so this is in 1. So 1 and 2. C1, C2, okay. So 1, 2. So it is between the C1 and C2, okay, of the ketonic group. So this is the structure of your sucrose. Okay, this is the structure of sucrose. The next is lactose, which is called as a milk sugar. It is called as what? Milk sugar. It is C12H22O11. Okay, it is made up of what? It is made up of beta D galactose. Okay, it is made of beta D galactose and beta D glucose. Okay, it is made up of beta D galactose and beta D glucose. So the structure is like this. Okay, the structure is like this. So this is one is galactose, another is the beta D glucose. Okay, so like this. So CH2OH, OH. So any will be asked any of this. So you have to practice this all. Okay, all the Hayward structures which I have drawn. So you have to practice it. So this is a okay. So here, so the linkage which is present. Okay, so you can see there is a linkage. So which type of linkage it is? It is a glycosidic linkage. Okay, it is a glycosidic linkage. So this is about disaccharide. Next, we are going to see the polysaccharide. So polysaccharide means what? It contains large number of monosaccharide units. So what do you mean by polysaccharide? So polysaccharide means it contains large number of monosaccharide unit. Okay. Yes. So, which are the examples of this your polysaccharide unit? So, first one is the starch. Okay, first one is the starch. So, this starch, what it is? It is called as a storage polysaccharide. Okay, it is a storage polysaccharide which is present in the plants. Okay, mainly in their roots. Okay, so it is mainly a polymer. Okay, which is made up of two this component, that is amylose and amylopeptine. Okay, you can see. So these are the polysaccharide components of starch. Okay, it is a polysaccharide component of starch which is called as amylose and amylopeptine. Okay, so this amylose, it is a water soluble. Okay, it water soluble and it consists of 15 to 20% of starch. Okay, and it is a straight chain compound. Okay, which is having 200 2000 alpha-D glucose units, which is held. Okay, how it is held? It is held by a glycosidic linkage. How it is held? It is held by a glycosidic linkage. Okay. Whereas when you are seeing the amylopeptine, okay, amylopeptine, it is insoluble. This amylopeptine, it is insoluble in water and it consists of 80 to 85% of your this what starch. Okay, and this is a branched polymer. This is what? This is a branched polymer. Okay, so amylose is a straight chain polymer and amylopeptine is a branch chain polymer. Okay, next is cellulose. Next is cellulose. So what do you mean by cellulose? So cellulose, it is also it is present in the plants. Okay, in the cell wall. Okay, and it is also a straight chain polysaccharide. Okay, which is consisting of beta D glucose units. Okay, it consists of beta D glucose units. Then we are going to see about the third, that is a glycogen. So glycogen, where it is used, it is mainly used as a storage or to reserve. Okay, carbohydrates, they are stored in this animal body. Okay, by which has glycogen. Okay, and body, it requires the glucose, glycogen, and it breaks down to glucose by the enzyme. So this is also called as the animal starch. Okay, and the structure of glycogen is similar to that of amylopeptine, which is highly branched. Okay, where this is present, it is present in liver, okay, muscles and brain. Mainly it is present here. Okay, yes. So what are the importance of carbohydrates? Yes, what are the importance of carbohydrates? So what are the importance of carbohydrates? So carbohydrates, they are the main source of energy, okay, in the organism. They are used for storage, okay, to store the molecules. For example, in plants like starch, and in animals, glycogen. Then there are insoluble carbohydrates. They act as a structural material like cellulose in plants, collagen in animals. Okay, and these carbohydrates, they provide raw materials. Okay, they provide the raw materials for the textile and paper, and they act as a lubricating agent in skeleton joint. Okay, they act as a lubricating this also. Then they are used in the formation of biosystems. Okay, in combination with many proteins and lipids. So this was about the carbohydrates. Next, we are going to discuss about proteins. So what are basically the proteins? So proteins are the essential biomolecule, okay, which is required for the growth and maintenance of the living organism. So these proteins, they are the polymers of what? What is the main what you can say, main component of proteins, of what makes the protein? So the polymer of alpha amino acid, right? So alpha amino acid. So when they are in the polymer form, so they make up the proteins. Okay, so proteins, okay, so they are the most abundant organic molecule of living system. They are important, okay, for the functioning of the cell. So and these, what are amino acids? So we said that proteins are the polymer of amino acid. Now, what are these amino acids? So amino acids, they are the compound which contains both amino and well as the carboxylic group. Right. So amino acids, they are the compounds which consist of what? Which consist of both the amino group. It consists of both the amino group as well as the carboxylic group. Okay, it consists of both the amino group as well as the carboxylic group in the same molecule. Okay, now depending upon the relative position of amino group and your carboxylic group, they are classified as alpha, beta, gamma, and delta, so on. Okay, so there, as we know, there are naturally available 20 amino acids are available. Okay, based on so there are 20 natural amino acids. So I can name some: glycine, aniline, valine, okay, then leucine, isoleucine, arginine, then your lysine, then you have glutamic acid, aspartic acid, glutamine, okay, asparagine, okay, then threonine, then serine, okay, cysteine, methionine, phenylalanine, then tyrosine, tryptophan, histidine, and proline. Okay, so these are the naturally occurring 20 amino acids. Okay, so these are the amino acids. So you can see here the structure of amino acid, right? You can see the structure of amino acid here at the right corner. Okay, it depends. Okay, it depends. Okay, so basically this how we can classify the amino acid. So there's something called as acidic amino acid, basic amino acid, and neutral amino acid. Now what do you mean by acidic? Acidic means it contains more number of COH. Okay, so it contains more number of COH group. That is, your carboxylic group are more than we called acidic. Okay, and which are the basic? Basic means it contains more amino group. Okay, so if I give an example of acidic, they are aspartic acid, glutamic acid, and your amino group, that is your basic. You can take lysine and glutamine. And neutral means what? Same number of or equal number of COH and amino group. Okay. So you can take the example as glycine and alanine. Okay. Now, based on the biological importance. Okay, biological importance. How important are these amino acids? So they are classified as essential amino acid and non-essential amino acid. Now, what do you mean by essential amino acid, students? Essential means what? Which cannot be synthesized in our body. Okay, and this has to be taken through the diet. Okay, whatever the food items we take, so through that we are supposed to what? Take it. So essential amino acids are what? Essential amino acids are they cannot be synthesized in our body. Okay, and have to take through the diet. And non-essential means what? They are present in the body. Okay. What are they? They can be synthesized by the body. Okay. So understood what are essential, what are non-essential. Very, very important. Essential means which cannot be synthesized in our body and has to be taken through diet. Non-essential means which can be synthesized by our body. Okay. So examples you have to give for both. Okay. Then um, what is the properties? What are the properties of amino acid? So the naturally amino acids, okay, all the naturally amino acids, they have L configuration. Then All the naturally occurring amino acids are optically active, only except one, that is glycine. Okay, because it contains the chiral center. Then these are amphoteric in nature. All these amino acids, they are amphoteric in nature. Okay. Then we are going to discuss about what are peptides and polypeptides. So peptide means what? An amide bond. There is a amide bond which is formed between the two amino acids, okay, with the elimination of water, okay, is called as a peptide bond. Understood? So what is peptide bond and amide bond? Amide bond which is formed between two amino acids with the elimination of water molecule. Okay, it is called as a peptide bond. So what do you mean by dipeptide? Dipeptide means it consists of two amino acids which are held together by a one peptide bond. Okay. Then similarly, tripeptide means what? Tripeptide means what? So in dipeptide, so number of peptide bonds, okay. In di will be one, in tri it will be two, okay. And in tetra will be three, and in penta will be four, like that. So it will be one less, okay. So chain having two amino acids linked by one peptide bond is called as a dipeptide. Now there is something called as polypeptide. What do you mean by polypeptide now? Polypeptide means when more than 10 amino acid, okay, when there is more than 10 amino acid and they undergo condensation, okay, then the product which is formed, it is called as a polypeptide. Okay, then the product which is formed is called as what? Polypeptide. Now, you know proteins. Okay, now you know proteins. Okay, so a polypeptide with more than 100 amino acids, okay, having molar mass more than 10,000 unified mass is called as a protein. So whenever a polypeptide, okay, that is a when 100 amino acids they are linked together, they form a polypeptide. Now this polypeptide, okay, residues which is having more mass, then they are called as a protein. Okay, so based on the molecular mass, the proteins are two types. One is a fibrous protein, another is the your globular protein. Okay, so when you see the fibrous protein, so these fibrous proteins means they are long parallel polypeptide chains. Okay, and these are like uh, there is a strong hydrogen and disulfide bonds, okay, which form which makes the fiber like structure, and these are insoluble in water. Okay, for example, the keratin which is present in the hair, then wool, okay, which is present in silk, then myosin which is present in muscles. So these are all examples of fibrous protein. Now what are globular proteins? Globular means these proteins have polypeptide chains, okay, and these are in coiled form. They are in coiled form, okay, and they give you a spherical shape, and these are usually soluble in water. For example, your albumin and insulin. Okay. Okay. So next we are going to see the structure of protein. So this is a primary structure. So what you can see in the primary structure. So basically there are four structure, that is primary, secondary, tertiary, and quaternary. Okay. Now what is a primary structure? So it refers to the linear sequence of amino acids. So like this. So linearly the amino acids are sequentially they are connected, okay, in the form of a chain, and they have a disulfide bridges. Okay, so this is a primary amino acid. Okay, so this primary structure is responsible for the function of protein. Okay, so the N terminal amino acid is written on the left side, whereas the C terminal amino acid is written on the right side. Okay, so like this. So N to C terminal will be present. So this is basically for the function of protein. Now this is a secondary. So secondary, so here the shape of the long polypeptide chain. So there are actually two different shape, that is alpha helix and beta pleated. Okay, alpha helix, see here how the alpha helix, what is done? The polypeptide chain is twisted. Okay, the first one, what you can see, the polypeptide chain is twisted into right-handed. Okay, by forming all possible intramolecular hydrogen bonds between the NH and the CO. So like this, the bonds are formed. Okay, and in beta pleated, what you can see, the polypeptide chains are stretched back. Okay, and out and fold method is taken here. Okay, and the chain is aligned side by side. Okay, and there is a strong intermolecular hydrogen bond. And how it is looking? It is looking like a pleated fold, right? So this is the secondary structure. Now tertiary, what you can see in tertiary? So in tertiary, you can see the 3D arrangement, right? You can see the 3D arrangement. So it gives rise to major proteins, that is the fibrous and the globular. Okay. Yes. Now quaternary, what you can see in quaternary, proteins are said to be quaternary in structure if they have two or more polypeptide chain. Okay, so hemoglobin is an excellent example. So, so some proteins are made up of two or more polypeptide chain. So then they are called as the quaternary. Okay, and so this is the example hemoglobin. You can take down the example of hemoglobin. Okay, yes. So what are the importance? What is the importance of the proteins? Okay, they perform the various functions, okay, as the enzyme, like antibodies, okay, as transport agents, okay, then biochemical messengers, they act as a structural materials, okay, etc. So these are the functions of the proteins. Okay. So now we are left with enzymes, vitamins, okay, and nucleic acids. So this we are going to discuss in the next class. Any doubt up to here? So what all things we have discussed today? Yes, students. So we have discussed what are biomolecules, right? What do you mean by biomolecules? We have discussed about what is biochemistry. Okay, then we have discussed about the carbohydrates. So we have seen what are carbohydrates. Okay, what is the classification of carbohydrates? So in monosaccharide, we have seen glucose. Okay, we have seen the structural elucidation. We have seen fructose. Okay, then we have seen in disaccharides, we have seen the maltose, lactose, okay, and sucrose. So these we have seen in disaccharide, and polysaccharide we have seen glucose, sorry, glycogen, starch, cellulose, okay. So these things we have discussed in the carbohydrates. Then we have seen about proteins. Okay, what are amino acids? Okay, what are the 20 natural amino acids? What are the types? Okay, so these all things we have discussed in amino acids. Then we have also discussed about the, what all things we have discussed again. We have discussed amino acids. Then we have discussed about the types of amino acids. Okay, nature of amino acid. Then we have discussed about the peptides. What are polypeptides? Then what are proteins? Okay, what are the structure of proteins? Primary, secondary, tertiary, quaternary, etc. Okay, so these are things which we have discussed in today's class. So in the next class, we are going to study about enzymes. What are enzymes? Vitamins and nucleic acid. Okay, is it clear? Fine. So I shall end this session here. So we'll meet in the next class. Thank you.